Photosynthesis is the process by which green plants make their own food. Green plants are called autotrophs. Now let's understand the meaning of the word photosynthesis. Let's break the word. Photo means light. Synthesis means to make complex substances from simple ones. The very word photo, which means light, tells us that light is the basic primary requirement for plants to manufacture their own food. Besides this, the raw materials necessary for photosynthesis are carbon dioxide and water, and only green parts of a plant like the leaves can photosynthesize, which means chlorophyll is also essential for photosynthesis. An important byproduct of the reaction is oxygen gas, which is evolved. There is an experiment which we will be doing to show that oxygen is evolved during photosynthesis. Now let us have a look at the equation of this anabolic process. Carbon dioxide reacting with water in the presence of light and chlorophyll, and this takes place in the cell organelle chloroplast to give us glucose, simple sugar, which would later polymerize to form starch. Water and oxygen gas is evolved. Now, the experiment we are going to perform is to prove that light is essential for photosynthesis because the radiated solar energy is trapped by the chloroplast and it is converted into chemical energy. You cannot perform this experiment at the spot. You have to prepare yourself for this experiment. To begin with, choose a potted plant whose leaves are medium sized. Don't choose plants whose leaves are very small. It could be medium sized leaves or even leaves which are bigger. Once you have chosen a potted plant, keep it in a dark room for at least 48 hours. That's about 2 days. The purpose of keeping this plant in a dark room is to de-starch it. After which, you choose few leaves. You take a black sheet of paper, cut that paper. Now this paper has to be very firmly fixed onto the chosen leaves, and I'll choose 2 leaves. I'll take U-pins to see that they are firmly adhered. I take another similar piece. I choose the leaf. In a similar fashion, I put the black piece of paper. I am now going to keep this plant in bright sunlight for a time period minimum of at least 3 hours and a maximum of 5 hours. It depends upon the intensity of light. If you have this Ganong's light screen, use it instead of this black sheet of paper. These pots, which were kept in a dark room for about 48 hours, have their leaves now completely de-starched. I select a few leaves, and to these selected leaves I have pinned black paper. I pinned it up tightly using these clips because these are going to be the leaves which I am going to test to show that light is necessary for photosynthesis. I have fixed the black paper for 2 leaves. Now this is yet another plant, but here the leaves are long. If you have in your lab the light screen, you can fix the light screen to the leaf, or you can use the same technique of fixing black paper. And now I am going to leave the pots in bright sunlight for at least 4 to 5 hours before I test them for starch. Now these are the leaves to which pieces of black paper were clipped. So the portion which was covered by the black paper should not answer the test for starch. I have brought the pot back now for testing. Remove these pins and the sheet of paper. Pluck the leaf and put it in boiling water. Now boil this for about 10 minutes. And meanwhile, as these leaves boil, let me tell you the apparatus we are using for this experiment. We have a beaker, the wire gauze, tripod stand, a source of heat, that is the spirit lamp, water bath. This is called as a water bath because we would be taking water in it to boil the leaf in alcohol. This is alcohol, iodine solution, another beaker, forceps and a glass rod. Also we would be making use of this petri dish, the black paper, scissors, the Ganong's light screen and also pins. Prepare yourself for the next step. Take another beaker, pour alcohol in it, now using your forceps, take out these leaves one by one and put them into alcohol. Now you have to prepare a water bath. Place the water bath on the tripod stand having the wire gauze and keep this alcohol containing beaker into the water bath, which means there is indirect heating because alcohol can catch fire. Now let this boil for some time. Remember, alcohol is a solvent for chlorophyll. Let's boil this for at least 10 minutes again. Alcohol is a solvent of chlorophyll. It is going to decolorize the leaves. Now, have a look. The alcohol has turned green in color. Let me pick up these leaves, take them out. You can find that they are totally decolorized. I hold it near this leaf. See the difference? I'll thoroughly wash it in water. I'll place it in the petri dish. And now I am going to use iodine solution to see which portion of the leaf answers the starch test. Now let me leave this aside for about 5 to 10 minutes and then observe the leaf. This is the leaf, boiled first in water, then in alcohol, washed in cold water, and I have placed it in the petri dish and I have put iodine solution which is brown in color. I leave this aside. You can see how successfully we have demonstrated light is essential for photosynthesis, because right the middle portion, right in the middle where a black paper was covering the leaf blade, no starch has been formed. That means the plant could not photosynthesize because light became a limiting factor. At the extreme ends, the iodine has turned blue-black, which shows that this portion which was exposed to sunlight has prepared food, which means it has photosynthesized. What are the precautions while conducting this test? You must place the potted plant in a dark room for at least 2 to 3 days for complete destarching of the leaves. Perform this experiment on a bright and sunny day. When you fix black paper, take care that it is firmly fixed, or else the results may not be very good. Take care to boil the leaves in alcohol, and alcohol should never be heated directly on the flame. The heating should be done in a water bath, otherwise it could be dangerous as alcohol can catch fire. After you have boiled the leaf, wash it in cold water, but please do not do it immediately because the leaf is quite soft when you have boiled it in alcohol. Cool it for some time before you transfer it to cold water, or else the leaf could break. In this case, we took care of all the precautions and you have seen the result is simply marvelous. Such an experiment can be done by changing the plant. Instead of having a plant whose leaves are fully green, you have a variegated plant, that is, the leaves have different colors in your crotons, your variegated leaves, and if you perform this experiment, you can also prove that chlorophyll is essential for photosynthesis. Enough, man.